Appreciate everyone joining today for this webinar focused on using soil biology to inform fertility decisions. The particular focus on BeCrop data, soil microbiome data, including Biomaker's newest offering, BeCrop Farm. I'm just going to check to make sure my audio and video is working properly, and I'll go ahead and kick things off. All right, so I think we're good to go. So as I've mentioned, thank you all for joining. This webinar titled From Soil Biology to Fertility Decisions, a Practical Approach to Improving Nutrient Use Efficiency, will really focus in on a particular section of the BeCrop offerings, which is nutrient cycling data. And not to get too far ahead of myself, but we have a lot of really cool practical examples of how to use BeCrop data, kind of connecting fertility-related inputs, biofertilizers, nutrient use efficiency enhancers, and really diving into some exciting updates as far as BeCrop Farm and BeCrop's recent improvements to some of the nutrient cycling indexes. So without further ado, we'll go ahead and quickly cover the agenda here. So we'll start with a quick intro and overview of BeCrop Farm, just for folks who may not be familiar with soil microbiome data and genomics. Then go over some of the fertility input products and tools on the market, just to kind of lay the groundwork for what sorts of inputs, management practices, and tools can be integrated into the decision-making process with BeCrop data. I'll give a really quick overview then on BeCrop nutrient cycling functional metrics, just because there have been some updates to make the data a little bit more practical, and kind of go over just briefly touch on the definitions of some of these metrics to kind of clarify what we're really looking at as far as soil microbiome nutrient cycling data. And then getting into the exciting part, integrating soil biology data to enhance nutrient use efficiency with a live demo where I'll be going over some data and just covering some examples of how this data can be used to make management decisions and understand how to unveil biological bottlenecks to nutrient uptake and ultimately crop yield. And then I'm going to try to leave at least 15 minutes or so for question and answers. On that note, some quick housekeeping items. Any questions that you do have as I go through the webinar, feel free to type into the chat, and I'll try to get to as many as I can in that last 15 minutes or so. And we'll also be recording this webinar and of course sending it out afterwards for anyone who maybe isn't able to stay on for the whole time or isn't available for the webinar. So yeah, excited to dive into this topic. And of course, I think it goes without saying that this nutrient use efficiency is a very relevant topic given the volatility in the fertilizer market and higher prices that growers are expecting to be paying this year, potentially next year. So obviously, as far as getting the most bang for the buck out of fertility products and also leveraging soil biology to get maximum returns on fertility is very important, very relevant topic. And we'll be, of course, touching on biofertilizers and a lot of the tools that you sort of expect to be associated with soil biology, but also talking about some nitrogen stabilizer products and some other nutrient use efficiency products and how those may be able to be informed using soil biology data. So a lot of cool stuff to cover and very relevant to a lot of the challenges growers are facing this year in the fertilizer market. So a quick introduction. My name is Gus Plowman. I'm a soil health and regenerative ag consultant that works alongside Biomakers. And my background is really focused on, I often like to use the term agroecology. So my background is really in looking at agricultural systems from an ecosystem perspective. So connecting the dots on the living things from microorganisms, bacteria and fungi to earthworms, protozoa, and of course, how that all relates to crops, and then the nutrient cycles and biochemical cycles that sort of connect those living components of farm ecosystem. So with my background, I've had experience leveraging soil biology testing tools and fertility tools that really take more of a biological lens than typical conventional farming tools and soil tests. So with that being said, I'll just give a quick overview of BeCrop technology for those who may be unfamiliar. BeCrop technology is developed by compiling a massive database of soil-borne microorganisms and then using advanced computing, AI-based technology to profile the functions of those microbes, which I'll get into the analysis process a little bit more in a minute. But really, the ultimate goal of this complex database and modeling technologies to provide actionable agronomic insights. So, obviously, there's a lot of really complex data that you can compile using genomics, whether that's plant genomics, human genomics, or soil genomics. But I think one of the challenges across all the different fields that use genomic data, including health, environmental science, and of course agriculture, is that data can be challenging to really put into a practical lens. And that's because when you're looking at genomics data, it can consist of massive lists of microbial taxa that even a really trained PhD expert is not going to be able to know off the top of their head what hundreds or even thousands of microbial species do. So ultimately, with BeCrop technology, the outcome is to provide actionable agronomic insights related to soil microbial functions, which I'll get into more in a minute. And BeCrop technology has been part of 34, maybe even more peer-reviewed publications. So then scientifically validated and leveraged in research related to soil biology, and has been applied across 6 continents and 56 plus countries. So like I said, really the goal here is to take really complex genomic data and put it into a practical lens to look at things like disease risks, stress adaptation and crop stress tolerance, nutrient cycling, which will be a major topic here today, measuring agricultural input performance, monitoring farm practices, measuring and verifying sustainability practices, and of course, providing the complex raw data on the soil microbiome, which with the right lens and expertise can of course be used in all sorts of really cool ways to better understand how the soil microbiome functions in agricultural systems. So now just a really quick rundown on how this data is actually generated. So, what a lot of people, I'd say for really a, vast majority of, modern farming history, going from maybe, the early 20th century or a little bit before that onwards to today, soil has kind of been looked at as an inert media or substrate that, holds some nutrients, and plants suck up those nutrients. But until recently, with the advent of regenerative ag movement and modern technology that allows us to actually measure and understand soil microbes, we now look at soil more as an ecosystem. It's not just inert dirt that sits in the ground and just kind of responds to physical and chemical factors, but also the biological aspect is deeply intertwined with those chemical and physical traits of the soil that historically have been more of a focus of agronomy. So with that ecosystem being made up of bacteria, fungi, archaea, protists, all sorts of microorganisms, there's a lot that that data on their populations can offer us, because of course those microbes are cycling nutrients. Some are posing threats as diseases. Some are able to suppress those diseases. Of course, there's a wide range of ecological roles those microbes play. So with the analysis process, I like to frame it as a 2-step process. First, like any typical conventional soil fertility test, starts with soil samples. Those samples get sent to a biomaker's lab or a partnership lab, and then the first step is that soil sample goes through an extraction process that extracts the DNA, and then that DNA gets sequenced to generate what you might refer to as biodata. So that's this biodata, this intermediate step here, is what generates the really complex data and like bunch of scientific names of bacteria, fungi, and other microbes, and of course their abundances. Like I said, that's hard to take into a practical framework for informing farm practices. So the second step in the process is really biomakers' bread and butter, where that biodata then goes through a complex machine learning based model that is able to generate a functional analysis report. So that essentially decodes what those microbes are doing in the soil. And gives you an idea of their functional potential. So based on their genome and how their sequencing data relates to model genomes and biomakers database, the models are able to generate the functional potential of that soil sample. So what extent of those microbes in the biodata are fixing nitrogen or solubilizing phosphorus. We'll get into the specifics more in a minute. So that's really a very simple rundown of the analysis process, but really it's the second step in the process here is really the important element that makes the data more practical and makes it possible to interpret from an agronomic lens. Really quick rundown on the data provided by BeCrop. So generally easiest to kind of break this down into three main categories. Soil quality here on the left, which is sort of the overarching look, sky down look at how the soil health status looks in general. So this includes metrics like soil biodiversity, how many different species are there and how even or abundant are they, overall functionality and resilience, fungi to bacteria ratio and mycorrhizal ratio. These are generally more informative indicators and are included on BeCrop test reports, but are not necessarily intended to be taken into account on as far as management practices, because generally these are, again, more top-down indicators of overall soil health status. They can be great for informing understanding how management practices are affecting the microbiome as a whole. But again, more of informative indicators to get an idea of outcomes like sustainability, biodiversity, and maybe what the overall balance of the soil microbiome looks in terms of these ratios. The plant health and nutrient cycling categories here in the middle and the right really dive deeper into the practical elements of soil genomic data. So this is getting down to those factors that ultimately can dictate crop productivity and yield. So these are really the major focuses, particularly of BeCrop Farm, which is designed to be essentially an in-field diagnostic tool for soil biology. So plant health section really includes anything that affects plant health outside of the nutrient cycling scope. So this includes soil-borne diseases, natural biocontrol agents, beneficial microbes that can suppress diseases, and then microbes that are phytohormone producers, basically microbes that produce plant growth regulators or PGRs, and stress adaptation metabolites, which is sort of a fancy term to basically mean microbes that are able to help crops deal with abiotic and biotic stressors. So you can think of that as like the microbes that are able to help crops deal with drought or heat or other extreme weather events, including indirect impacts from diseases. And then on the right side, we have nutrient cycling, which will be the primary focus of this webinar. This includes categorizing microbes and measuring them based on the extent to which they're able to cycle nitrogen, phosphorus, and potassium. And of course, there's a lot of different roles microbes play in each of these nutrient cycles. So it's not just one specific set of microbes, but several different functional groups within each of those particular macronutrient pathways. And then carbon pathways, which as a soil health advocate, I would be remiss if I didn't touch on carbon pathways a little bit. So we'll definitely be diving into that, even though nutrient cycling will be a primary focus. Within carbon pathways, one important metric is organic matter breakdown or decomposition, basically the microbes that are breaking down crop stubble and residue and other organic materials that find their way into agricultural soil, like insect carcasses, manures, and compost. So obviously this also relates to nutrient cycling and will certainly be something that we touch on. And then micronutrient cycling pathways. So the microbes that are able to unlock some of those micronutrients that can be challenging, like looking at like manganese and sulfur and just some of those. Obviously there's some different perspectives on what's categorized as a macronutrient, micronutrient, but really this is just looking at some of those minor elements that plants don't typically need in quite as large of quantities as NP and K. And then additionally, BeCrop testing also offers optional chemical fertility tests through partnership labs, including the Haney soil health test, which gives a great biological lens to soil fertility and pairs really well with BeCrop testing to give a broader perspective on both the nutrient availability, soil respiration, and then of course the soil genomics data sort of being the mechanisms behind some of those nutrient pools that the Haney test measures. And then also the Malick, standard Malick tests also, of course, pair well to get an idea of both the chemical nutrient availability as well as the biological nutrient cycling, and total soil digestion to measure the overall level of sort of the backbone or the savings account of nutrients in the soil, whether or not measuring every every nutrient, whether or not it's available to plant uptake or locked up in the soil. So as far as BioMaker's solutions, there's really three main offerings, including BeCrop Test, individual soil tests, which are a great sort of a standard snapshot of soil health status with all the metrics we just went over. BeCrop Farm, which goes quite a bit deeper with AI-driven agronomy insights on a precision ag level. So a mapping tool that allows you to really go beyond just a single test, but to look at data across acreage and on a much higher resolution scale. And then BeCrop Trials that measures the impact of inputs on the soil microbiome. So really more of an R&D and product assessment tool to hone in on the specific changes in the microbiome, the benefits of input products like biostimulants, fertilizers, crop protection products. And now a little rundown on BioMaker's newest offering, BeCrop Farm. So BeCrop Farm, as I mentioned, provides what I would define as precision ag data on soil biology. And the way this works is by integrating a wide range of remote sensing data sets. So think like NDVI, which measures essentially the levels of plant biomass in the field, soil texture, soil moisture, and a whole large suite of other factors as well that can be measured using remote sensing data from satellites and such. And then it pairs that with targeted soil tests from representative zones. So it uses that remote sensing data, which we know that NDVI, soil texture, soil moisture, all these factors, just like the biology can have influence on the soil, physical and chemical components. These components of the soil also can provide us with an understanding of how the soil microbiome varies across the field. So pairing those remote sensing data points, BeCrop Farm was developed to present or basically calculate representative zones across the field so that we have an idea of how much soil biology variability there is. And then that data, when physical soil tests are taken and paired with that environmental data, it delivers these precision soil biology maps, kind of like a heat map across the field to denote really how the levels of certain biological functional groups vary. Really just a quick rundown on this process with the sampling protocol. It starts with shape files of the field because that's needed to understand, the extent of the, geographic area that the satellite-based and remote sensing data will look at. So first, shape files are uploaded, and then those environmental variables are looked at that influence soil biology to determine what specific biological zones look like, how much variability is there in the field, is it really homogeneous, really uniform, or is there a lot of differences across the field in terms of soil biology? And that determines the sampling location so that folks can go out, and instead of having to pull a sample every 1 acre or 2.5 acres or 5 acres like a typical grid sampling protocol. In this case, those biological zones, rather than doing that high intensity sampling, only a handful of samples will be required from those representative zones to provide an accurate map and get an idea of how soil biology varies across the field. So after the sampling zones are determined, coordinates are sent back to the client for sample collection. And then of course, once the samples are submitted, that's when the model ultimately runs and computes the output data, basically like a precision ag map, similar in many ways to a grid sampling map, but with less sampling intensity and labor involved. So I think I got a little bit ahead of myself, but this is basically that process broken down in simple format. Soil samples are taken, you have these field level zones determined based on soil biology variability. And then this also can be integrated on a whole farm area or across multiple clients or regional field views for, say, a retailer can look across multiple farm operations and look at fields within those farm operations. So a little bit of a hierarchy as far as organizing the data to put together harmony between whoever's taking the samples and whoever's interpreting the results. And of course, for a retailer, those results for an individual farmer or client can be shared with them, through the BioMaker's BeCrop portal, thanks to this data organization system housed in the portal for BeCrop Farm. So now to get into our webinar topic today, I just wanted to set the tone by quickly going over agricultural, fertility products and practices on the market to get an idea of the types of solutions that BeCrop Farm can inform. This is by no means a comprehensive list. This is really, I think, just a list of some of the well-known and understood fertility products and tools on the market, but I'm sure there's dozens of solutions that don't fit within some of these categories, and these are also fairly broad. So as far as fertility inputs, of course, you have your conventional fertilizers that go out on a vast majority of acres, particularly in row crops. And then enhanced efficiency fertilizers, think of like polymer-coated fertilizers or fertilizers that may be formulated with, say, humates, humic acids and such to improve, to basically make them slow release or make them more biologically available. Foliar fertilizers, which are a solution that, of course, can sort of bypass that soil environment and put the nutrients right on the plant foliage for direct absorption to be higher efficiency. Organic fertilizers and soil amendments, so your compost, manure, fish hydrolysates, all sorts of great organic fertilizers out there that provide fertility maybe at lower concentrations than conventional fertilizers, but often are more biologically available, slower release, and also able to stimulate soil, essentially feed soil microbes in addition to providing nutrients that ultimately are available to the crop. Biofertilizers and biostimulants, which we'll be talking about quite a bit, thinking like microbial inoculants, other products out there like humic acids and certain plant extract-based products that are known to improve nutrient cycling, either by feeding the microbes, stimulating them, or by introducing beneficial nutrient cycling microbes. I would throw maybe mycorrhizae inoculants as one example of a product out there on the market that gets tossed into the biostimulant or biofertilizer category thanks to mycorrhizae's ability to help allocate nutrients to crops. And then nitrogen stabilizers, another one that might not be considered biological, but of course, when you have nitrogen being so volatile, there's a lot of products out there that fall in the nitrogen stabilizer category, like urease inhibitors that can slow down those processes, including biological processes that lead to nitrogen loss from the soil. And then as far as fertility tools, another element to take into the equation as far as informing management practices with soil biology data. So in-furrow applicators and side dress applicators to really follow those 4R protocols of right place at right time to be able to apply fertility products right in the root zone and at the time when crops need them, in the case of like Y-drop and in-season side dress. Fertigation systems that apply nutrients through irrigation, variable rate technology for precision ag, water treatment systems that maybe if you look at soil biology data and see concerns as far as maybe looking at things like salinity and stress, and that there's issues as far as, say, irrigation water salinity, water treatment systems is one tool that can be actually pretty vital to maximizing nutrient use efficiency, and then soil sensors and probes. So we probably won't touch quite as much on fertility tools, just given that fertility inputs are maybe a little bit more directly applicable, but many of these fertility tools definitely should be part of the conversation as far as understanding how soil biological data can be used to maximize nutrient use efficiency and inform management practices. So a really quick overview of soil biology nutrient cycling processes. These are just general examples of diagrams of a nitrogen cycling and phosphorus cycling processes that go on throughout the soil. And this doesn't, these are not comprehensive diagrams, again, just examples, but this just sort of gives a demonstration of of all the roles soil microbes play in converting nitrogen and phosphorus between different forms, making it plant available, and also causing loss of these nutrients from the soil environment. So for example, as far as nitrogen, you have your nitrogen fixers that can live either in legume root nodules or be free living, that play a role in importing nitrogen from atmospheric sources. Then, of course, you have microbes that drive the conversion of nitrogen from various forms, from organic nitrogen or nitrogen that may come from nitrogen fixation to convert it into ammonia through mineralization and nitrification that further moves that nitrogen from the ammonia form to nitrites and ultimately nitrates, which makes those nutrients potentially more available to most crops, but also makes them more liable to being lost from the soil through leaching and processes like denitrification, which is another microbial driven process that we'll touch on here. And then phosphorus, I don't need to go through the intricacies of the phosphorus cycle, but of course, every nutrient cycle is different. And likewise, microbes play a wide range of different roles. In the case of phosphorus, microbes make it more available by solubilizing it or unlocking it from mineral forms, and also converting it from organic forms through the process of mineralization. So of course, these nutrient cycles are much more convoluted than what these diagrams are sort of simplifications, but they give you an idea of really how much complexity is there, and of course how with BeCrop Farm we're sort of diving into the really key elements that can affect crop uptake and availability of these nutrients. And that includes metrics that both positively and negatively influence bioavailability of nutrients. So there have been some updates with the recent BeCrop 4.0 release as far as nutrient cycling, and I wanted to touch on those for folks who may have been using BeCrop for a really long time. And some of these updates, particularly as far as nitrogen cycling, make the data quite a bit more practical as far as understanding how it can carry over into making input decisions. So first of all, sort of the headliners of the nutrient cycling category that we'll be touching on is our global NP and K nutrient cycling metrics. So you can think of these like the overall biological nutrient cycling pathways. So there's a global nitrogen, global phosphorus, global potassium index, and these are basically a representation of this overall nutrient cycle like we just went over here on the previous slide. So these integrate all the different roles microbes play in cycling these nutrients and provide an overall score, with a higher score indicating greater microbial potential to make these nutrients available. So it's really, again, just sort of a way to give an overall benchmark on what the nutrient cycling structure of the microbes looks like. But then, of course, when we get into specifics, there's all these much more specific roles that microbes play in terms of converting nutrients from various forms. So I broadly categorize these into two different categories: macronutrient gain and supply pathways, basically the processes that microbes perform that unlock nutrients. And this includes what we call gain of nitrogen to ammonia, or nitrogen mineralization, basically unlocking that nitrogen, converting it to ammonia as microbes cycle it. And then gain of nitrogen to nitrate, nitrification, sort of an intermediate process in the nitrogen cycle that releases nitrate. Gain of nitrogen from gas, otherwise known as nitrogen fixation, obviously a really big focus of a lot of biological inoculants and beneficial microbial products. And then outside of the nitrogen scope, which those nitrogen pathways are really part of the major update with BeCrop 4.0, and then phosphorus solubilization, the major supply pathway in which microbes unlock phosphorus, and potassium export, the process whereby microbes are basically releasing potassium in bioavailable forms for crop uptake. And then on the flip side, we have macronutrient loss and competition pathways, which reduce nutrient availability for plant uptake. This includes loss of nitrogen to biomass, also known as nitrogen immobilization, whereby microbes themselves actually consume nitrogen and potentially compete with the crop for it and convert it into their own biomass, which makes it less available to the crop in the short term. But of course, there are some mechanisms where microbes, through like rhizophagy, a process where plants absorb microbes directly, that immobilized nitrogen may still make its way into the crop. So it's not necessarily the same as nutrient loss. But then we do have loss of nitrogen to gas, denitrification, whereby nitrogen is released back into the atmosphere from the soil. And then loss of urea by volatilization. Just really the term volatilization gets thrown around a lot to mean nitrogen loss, typically from urea fertilizer. And then phosphorus and potassium consumption, basically the same thing as immobilization, where microbes consume phosphorus and potassium for their own use and allocate it to their own biomass, which again doesn't mean it's lost from the soil, but means that in the short term it may be locked up in essentially the organic matter or in the microbes. But of course, as microbes, they could potentially go through some processes where those, maybe they're absorbed by the plant through rhizophagy, or perhaps as those microbes die off and their populations turn over, or they get consumed by a protozoa, some of that phosphorus, potassium, or nitrogen can then make its way back into plant available forms. And then lastly, the micronutrient cycling functions, which do all go by different names, but these basically just represent kind of like the global NP and K scores, the overall potential for microbes to unlock nutrients like iron, zinc, magnesium, calcium, manganese, sulfur, copper, and chloride or chlorine. And I know that some of these are sometimes categorized as macronutrients, but for the purposes of the BeCrop report, these are fitted in under NPK as micronutrients. So now to get into the fun part, I'm going to do a quick live demo integrating soil biology data to better understand how to inform fertility management and nutrient use efficiency. So for this, I'm going to jump out of my slide deck here and into the BeCrop portal. And I have a project pulled up we're really gonna drill into how some of the results from a BeCrop Farm project can be used to make decisions. This is going to be sort of a generic use case. This is actual data, but of course, as far as outcomes and decisions, the specifics are really going to depend on your context. So again, this is sort of general guidance, but to drill deeper, it may depend a lot on what tools are available, the cropping system, whether or not you have irrigation. So in this case, we're looking at center pivot cornfields. And with BeCrop Farm, the portal here kind of gives you the scores on this toolbar on the left. And it starts with those sort of, like I said, headline metrics, including those global NP and K cycles that represent the overall microbial ability to make these nutrients more available to the crop. I'm just going to center in for now on these 3 particular center pivot fields, and kind of give an overview going top to bottom on what some of this nutrient cycling data can tell us as far as decision making. So first of all, a little refresher on how the BeCrop scoring system works. Down here in the bottom right, you can see a scoring scale that ranges from very high in the dark blue to very low in the bright yellow. This is based on a 0 to 100 index that ranks all of the samples in Biomaker's database for a specific crop type. So in this case, corn, and it ranks those samples according to the levels of microbes that support various functions. So each metric will have a score 0 to 100, with scores of 100 meaning that the sample ranks, or the field ranks at the top of the database, and a score from say 0 to 20 falls in that very low category, indicating that that particular field area ranks in the bottom of the index. And then in the middle range, you have medium, which is about the 40 to 60th percentile range, which would mean that soil roughly looks about average in comparison to the database. And then you can also see there's a tool here, since this is not scored on by sample basis, but on an area basis or acreage basis. You can toggle here between acreage and percent area to see how many acres or what percentage of the farm falls under each of those scoring categories for a particular metric. So in this particular case, first of all, with BeCrop Farm, what it immediately highlights are potential metrics to improve here in the results toolbar. So any metric that falls, well up, or I should say, any agronomically vital metric. So this potential metrics to improve section really prioritizes the metrics that we know relate to important crop-related factors that can be bottlenecks to yield. So think of like macronutrient cycling pathways, major diseases, things like that we know just from a basic agronomic philosophy can be significant hurdles to crop productivity. So in this case, what stands out is the global potassium index. So again, that's looking at the overall microbial ability to cycle and unlock potassium, and that scored 25 out of 100. So sort of in that lower tier of the Biomaker's database with a score of 25. And this when we look at the field, we can also see sort of the variability here, and click on each field to get an idea of what the score is. So they're all in the yellow, they're all in the very low to low range, with a few little blue specks here that I think are in the medium range. But when you click on a field here, you can see for this one, field 3 in the top, the average score was 31, so a little bit higher than the farm average here on the left of 25, but a majority of that's still in that low category, about 52 acres, which looks like it's majority range. And then a couple of these fields scored even a little bit lower, score of 23, and I think this was the lowest, this bottom right field, with a score of 19. So a little bit of variability, but overall across these fields, so a lot of room for improvement in terms of potassium cycling. And of course, as far as solutions go, just from a general perspective, products that make claims of improving potassium use efficiency or potassium cycling would definitely be options here. But as we go down into the specific nutrient cycling metrics, we can get a little bit deeper into diagnosing what's going on here and even tailor that a little bit further. But really, the headline metrics here just give you an idea of what nutrient cycling, which specific nutrients may have cycling issues, with potassium being the big challenge. Nitrogen, phosphorus all scored pretty high. We see nitrogen scored really high in this top field with a lot of dark blue. This bottom right field scored the lowest with some sort of hot spots in the yellow. That could be spots that maybe you could do a targeted solution to improve nitrogen cycling. But again, this is looking at the overall nitrogen cycling picture. So, this may sort of give you general information on, prioritizing. If you were looking at just these global nutrient cycles, I would look for potassium related products, enhanced efficiency fertilizers, biofertilizers, things like that that make claims for potassium. Maybe like green sand, which is a mineral product that contains potassium in forms that microbes are able to cycle, and is lower salt index fertilizer than a lot of conventional NPK fertilizers. But then, of course, to get down into specifics, we go down to the all metrics section and click on nutrition here. And this is where we really dive into the specific roles microbes play in each of these nutrient cycling categories. Really, these are kind of what fits into some of those global nutrient cycling indexes. So just from top to bottom, gain of nitrogen to nitrate. This is, again, the nitrifying microbes, sort of those intermediate steps. And this is an interesting category because you always need to rely, as far as nitrification, there's generally for to supply nitrogen to the crops at the pace that they need it. It's great to have some nitrification going on. There's typically going to be nitrification in soils, particularly when it's warm enough and there's enough moisture and the pH is generally within reasonable range. But of course, if you have nitrification too high, microbes are going to be driving that conversion of nitrogen to nitrate so quickly that a lot of that nitrate may be volatilized or leach out of the soil before plants can uptake it. So in this case, it's on the lower end. So, this may be a case where, of course, temperature and soil chemical factors like pH can play a role. But generally, when you're able to adequately manage soil moisture, have, oxygen availability, nitrification, generally, ideally would sort of be in the intermediate range, not too low, not too high. This was a little bit on the lower end, but one of those cases where maybe it could be sped up a little bit through, nitrogen products that may contain organic forms of nitrogen, which would kind of stack up nitrogen ahead of the nitrification process. Whereas this would not necessarily, if you have really high nitrification, unlike this example, you would not really want to be applying a ton of, ammonia nitrate-based fertilizer, because then those nitrifiers are going to convert it potentially very quickly, convert that ammonia into the nitrate form. And then you're going to have so much nitrate that, it may be in excess of what the plants need. And a lot of that nitrate is just going to be lost to the environment or runoff through water, irrigation, and rainfall. Loss of nitrogen to biomass here. This is basically measures microbes that immobilize nitrogen or uptake it. And this also can be dictated by environmental factors. But really, if this can be high, particularly when over-fertilization is a problem. So when fields regularly get high rates of conventional inorganic nitrogen forms like ammonia and nitrate, the microbes themselves, it'll select for the microbes that do immobilize those nutrients, and for lack of a better term, sort of make the other nitrogen cyclers lazy, because they can just rely on those synthetic forms of nitrogen to get it. So that can spike this level when there are large applications of those synthetic inorganic nitrogen fertilizers. In this case, nitrogen immobilization is low, which is sort of what you want to see, indicating that the microbes aren't necessarily stealing or competing with the crop for that nitrogen. Gain of nitrogen to ammonia. So this is looking at nitrogen mineralization, which is that beginning process where microbes convert organic nitrogen into ammonia. Generally, a score of 51 here is fairly solid. If you have really high mineralization, kind of like nitrification, that can indicate that microbes are moving the nitrogen, cycling it too quickly down the pathways, and then a lot of it's going to get lost. Not as much of it's going to make it into the crop. But to make use of organic forms of nitrogen and say compost, fish hydrolysates, other organic forms, it's very important to have some of these mineralizing microbes. And if this score is low, like in this case, it's in the medium range, but there are some hot spots in a couple of these fields we see in the low ranges. And this is where maybe applying organic forms of nitrogen, like I mentioned, maybe fish hydrolysates, corn steep liquor, a lot of products out there that are basically organic nitrogen fertilizers that can potentially give microbes some of this organic nitrogen to work with and convert to ammonia. And then we have some loss pathways, loss of nitrogen to gas or denitrification, and loss of urea by volatilization. And really, this is where nitrogen stabilizers can play a big role in our fairly industry standard product as far as slowing down some of these nitrogen loss pathways. So this is a case where this particular case study does look like a great prospect to maybe use a nitrogen stabilizer. And when I say nitrogen stabilizer, that can include urease inhibitors and as well as maybe organic or natural nitrogen stabilizers or chelating agents like humic acids are looked at as one, amino acid products. Those are also compounds that can potentially hold on and bind nitrogen, reduce its loss, and prevent some of the challenges that these biological pathways can have to reducing nitrogen use efficiency. So this would be a great candidate for, say, humics, nitrogen stabilizers, amino acids, other products that may be referred to as biopolymers or chelating agents to hold that nitrogen in the soil. And that could probably be said the same for loss of nitrogen to biomass. Really, those products that make claims of helping improve nitrogen stability, that's where these loss pathways are really a great insight on making decisions regarding those products. And then we have another gain pathway, gain of nitrogen to gas or nitrogen fixers. So in this case, it's particularly high in these fields. And interestingly, some of the nitrogen fixation can be driven down by excessive applications of inorganic fertilizers, just like I mentioned that loss of nitrogen to biomass or immobilization can be driven up. But in this case, with this field, we see really what you want to see, which is low immobilization, not quite as, obviously some immobilization is good because it does help hold nitrogen in the soil, but you don't want microbes uptaking so much nitrogen that the plant can't get enough. And you also don't want microbes relying on inorganic inputs instead of producing some of their own nitrogen or supplying it as through nitrogen fixation. So if this is low gain of nitrogen from gas, then of course it's a great candidate to maybe look at integrating legumes into a rotation to help select for some of those rhizobium and other legume associated nitrogen fixers. And of course, there's a lot of free living nitrogen fixing products on the market, but also just ensuring adequate soil management, reasonable soil moisture, eliminating compaction issues can also naturally help create an environment that can cultivate these nitrogen fixers. And then to go into I see we're already 15 minutes, so I'll try to speed things up. A lot to get through and a lot to unpack as far as nutrient cycling. But now to go beyond some of those nitrogen indexes into phosphorus solubilization, primary pathway whereby microbes are unlocking phosphorus from bound up inorganic forms in the soil. So this scored quite high. So in this case, not really a good example of where you might use this index to make decisions, but particularly if inorganic phosphorus solubilization is low, that's where you may apply, say, a phosphorus biofertilizer that contains organic acids that can unlock phosphorus or contains microbes that are themselves phosphorus solubilizers and can sort of augment this category as far as the levels of these microbes. And then phosphorus consumption, just like nitrogen, we have microbes that can unlock or basically consume phosphorus and compete with the plant for it. So that scored low here, which obviously, you don't want to see phosphorus consumption really high because then, of course, microbes can be competing with the crop for phosphorus. But just like nitrogen, excessive inorganic phosphorus fertilizers can drive this up because it can select for microbes that consume phosphorus instead of solubilizing it and unlocking it on their own. in this case, we see sort of a more favorable balance with high phosphorus solubilization and low phosphorus consumption. Now to get to what I was looking forward to, which is the potassium indexes. So as far as supply, we have potassium export, which are basically the microbes that are releasing potassium from their cells in bioavailable forms. And that scored low, about a score on the 0 to 100 range of 20. And then potassium consumption, whereby microbes are immobilizing potassium, consuming it themselves. This scored quite high with a score of 72. And we see some variability with this field scoring 66 on the top and this field in the bottom right scoring all the way at 80. So this particular field in the bottom right looks like maybe it has, and I'm totally speculating, this is just a general example, but if you have high levels of potassium, inorganic potassium fertilizer being applied year after year, just like nitrogen and phosphorus, that can select for high levels of those microbes that consume potassium and rely on the fertilizer input rather than naturally cycling potassium and exporting it to the crop. So another piece of this, which I touched on the BeCrop Plus test earlier, which integrates chemical fertility data, but that's included here in the top in this physical chemical tab. And remember, when we're looking at this field 1 here has high levels of potassium consuming microbes, and then this field in the top a little bit lower, so a little bit more favorable. But then with the physical chemical data here, you can sort of look at what that pool of nutrients looks like and get an idea of what the relationship might be between the nutrient cycling levels of microbes and the actual level of available phosphorus, potassium, and such. So I'm navigating to available potassium, and remember it was this field in the bottom right which had these the highest levels of potassium consuming microbes. So this scale basically goes, this scale goes from dark representing low to the light areas representing high. It's just based on the range within these fields. This is not based on the Biomakers database or anything like that. We can see that field 1 here, which had high potassium consumption, has low available potassium compared to the other fields, with about 170 pounds per acre compared to 206 and 208 in the other fields. So again, this is a generic case study and sort of speculatory, but it looks like this field 1, you are seeing lower levels of available potassium. And to dive deeper, because obviously there can be a lot of reasons you have lack of potassium. It could be getting locked up. It could be that there aren't enough microbes sort of making it bioavailable. But going back to the nutrition section, this does line up with what we're seeing as far as potassium consumption. Seems that there's excessive levels of these microbes consuming potassium here that may be leading to lower levels of the available potassium on the soil fertility report that we just looked at. So this would certainly be a candidate based on this data to say, look, maybe instead of applying inorganic potassium fertilizer, you look at converting some of that to say, forms like green sand is a good example. Again, a mineral potassium product that supplies potassium in a form that microbes can cycle more effectively, or say, potassium from organic forms like manures or compost or bat guano, all sorts of forms of soil amendments and inputs out there that can supply potassium in forms that encourages microbes to cycle it and unlock it a little bit more, rather than encouraging them to just go ahead and consume it like those inorganic forms. So there's a lot to unpack in the nutrient section, but I want to jump into questions here. And there's a few things I didn't get to, but I did want to touch on the micronutrients and a solution that can be used here with some of these micronutrient pathways. So looking at the toolbar here, it looks like iron, copper, manganese, and magnesium scored in the lower ranges, whereas chloride, zinc, sulfur, and calcium scored pretty high. So these, zinc, sulfur, and calcium in particular, looks like there's high populations of microbes that are able to unlock these micronutrients. But iron, manganese, and magnesium could be problems. And again, I'm not going to do it for the sake of time, but you can look at the physical chemical data here if you opt for BeCrop Plus testing and can see, what the available levels of these micronutrients are, which is another important factor. But say you do have iron deficiencies or copper deficiencies or low levels of, available iron, copper, manganese in the soil, and you do seem to have these low levels of microbes cycling these nutrients. Obviously, there aren't a ton of products out there that necessarily make claims of improving nutrient cycling for these micronutrients. But as you integrate cover crops, compost, some of these soil health management practices that can sort of just improve soil health as a whole. That can certainly boost micronutrient cycling. And then there are some tailored products out there that may contain chelated forms or complex forms of these micronutrients, which can be really vital to just make sure that these that basically chelated or complex nutrients just provide these micronutrients in forms that make them more available, more stable for the crop to uptake in the soil profile. And then one last one, which I think is a really important tool that's been around for a little while, but does get talk from a lot of soil health experts, are foliar fertilizers. Obviously, with foliar fertilizers, you may not be necessarily improving nutrient cycling in the soil directly, but you can help bypass some of these bottlenecks to unlocking these nutrients by applying, say, a foliar, iron or manganese or magnesium. And of course, there's a lot of foliar products out there. Some are going to maybe integrate multiple micronutrients. Some are going to pinpoint specific micronutrients. But that's a great, obvious, with foliars, you want to be thinking a little bit about crop tissue tests and maybe what deficiencies you're seeing pop up. But to look proactively, even before the season, and be able to see, look, the biological population does not look strong enough to be constantly cycling some of these micros. That's a case where foliars can be a great tool to help ensure that the lack of these micronutrient cyclers doesn't lead to deficiencies later on in the season. One thing I really wanted to touch on that I didn't quite have time for is the carbon pathways. But as a soil health advocate, I definitely have to touch on it really briefly. But there is a suite of these carbon metrics here related to how microbes are importing carbon through processes like carbon fixation, and organic compounds degradation here is decomposition. These obviously are big processes for importing carbon into the soil, or basically taking, sorry, I have a truck out in front. Okay, organic compounds degradation, like microbes that drive decomposition, are really crucial to breaking down crop residue and stubble, unlocking nutrients and sequestering some of that carbon, basically stabilizing those organic inputs and crop residue into organic matter. So in this case, this scored really well in terms of decomposition or organic compounds degradation, these three fields. But if this was scoring low, that might be a case where applying a carbon-based biostimulant could potentially jumpstart that and help get those microbes cycling these organic compounds and breaking them down. Or another important factor to think about that relates to the physical chemical metrics is looking at your carbon to nitrogen ratio. If you have an imbalanced carbon to nitrogen ratio, that can certainly affect these populations. And then lastly, to have a there's a lot of products out there that are residue decomposer products that contain microbes that are able to basically fulfill this function and perform this service of breaking down crop residue. So that would be a great solution if this metric scored in the lower ranges. Carbon-based biostimulant or a crop residue decomposer, stubble decomposer product is what can help you get a lot of potential ROI out of those nutrients in that biomass or in compost and such. There's a lot to get through here. I hope that provided some insights as far as how this data can be used in practical scenarios to inform some of those input decisions. And then also what I didn't touch on as much are some of the tools like in-season applications, side dressing, Y-drop, and things like that. But that certainly serves as another tool, kind of like foliar, to help provide the nutrient when the plant needs it, where it needs it. Where if you have deficiencies, and this could be applied to the global potassium cycle or global nitrogen cycle if that's scored low, where you can provide that nutrient in a form right there in the root zone where crops can uptake it. So some of those precision 4R, nutrient placement and in-season application processes can be an awesome way, to potentially address biological deficiencies in nutrient cyclers. But some of the initial recommendations I went over, like humics, biostimulants, amino acids, organic fertilizers, those are really great solutions to actually improve some of the root causes of these issues and augment these microbial populations. All right, so I'm going to cut myself off here and dive into some questions. And let's see. Okay, first question. Can you explain AI-driven bioinformatic technology, how it defines the microbes functioning? Yeah, and that might be a good question to go back to my initial slides for this one and dive a little bit into this process here. So hopefully my slide deck is popping back up here as I share my screen. So this is, again, a very simple, interpretation of the process, but basically with the bioinformatic technology, we're referring to this second step where the biodata, and again, this biodata is like the complex, the species and genus level information on soil microbes and their abundances. And then the AI-driven bioinformatics process takes that and runs it through a massive modeling data set that includes reference genomes. So basically, the Biomakers data science team has spent years compiling these genomes of microbes that allows, with machine learning, to infer what the microbes in this biodata, what their roles are in the soil. And the way that essentially works, you can the simplest way I like to think about it is it's sort of like reconstructing phylogenetic trees, or basically, I guess a simpler way of putting it is if you have a microbe in the reference database that is, say, an ancestor or distant relative of microbes that are detected. It's kind of like if your parents or your grandparents did 23andMe or did a genetic profiling test, that could be used to predict and know what your eye color is, because through your ancestors, through your parents, grandparents, or even further back, you can see what their genetic profile is, and then that can be used to determine your eye color, other disease, genetic risk factors as far as disease. So that's it's not exactly like that, but that's similar to how this bioinformatic process works. Is it uses reference genomes, and then those are used in a model to determine what the functions of these microbes that are sequenced and detected in the soil sample actually do in the soil. One really cool aspect of this process is that many microbes out there are unknown or basically unnamed, haven't been cultured in a lab, so they haven't been given a name. But if you have microbes that are unnamed and relatively little is known about them, if the Biomakers reference database has ancestors of them where you can see that they have some similarities in their biomarkers, the model, like I said, just like if you have your parents do 23andMe and you can determine what your eye color is without actually looking at what your eyes look like. It works the same way here, where even an unknown microbe, or maybe a microbe that doesn't have a species name but fits within a genus, using those reference genomes, the bioinformatics process can profile what functions those microbes perform without them even being, maybe a named microbe. So it definitely goes a lot into a deeper rabbit hole to get, and definitely beyond my scope of knowledge as far as how that bioinformatic process works. But that's the gist of it. And this process is referred to as amplicon sequencing or metabarcoding, as far as how those microbes are identified. But then this bioinformatics process, the second step, really is what provides the major value as far as practical data. Another question, how many soil tests are needed in a circle or in a center pivot to provide this level of detail? Are the soil tests benchmarked to imagery, or is the report all based from the soil tests? So the first part of that question, how many soil tests are needed, that actually varies depending on the variability within the field. So I guess maybe this is an example here of this picture where there's a lot of variability, as indicated by all these different colors showing basically different biological zones based on that satellite data. But generally, I can't give an exact number for a center pivot field. Depending on size, obviously you have a bigger field, you may have more samples, but it depends on how much variability there is. So it may be as few as 1 or 2 samples, maybe a little bit more if there's a ton of variability, but generally it's not going to be much more than that. So it's really the way that the process works is it determines those biological zones to minimize the number of samples taken. But yeah, it's totally, that's why a shapefile needs to be submitted, so that aerial imagery data can be, or satellite data can be used to determine how many samples actually are needed. And then, are the soil tests benchmarked to imagery, or is the report all based from the soil tests? It integrates both. So it uses the soil tests as well as that remote sensing data to basically predict and extrapolate out what these levels of microbes look like across the field. So the answer there is both, and that's a big part of the value with BeCrop Farm. And I know we're at the top of the hour, so maybe one other quick question. Let's see. Could you please clarify which particular molecular tests you're using? That would be amplicon sequencing or metabarcode sequencing are the sort of the industry standard names for those. And for anyone else who's submitted questions, we'll make sure to follow up with you and get you some answers over email. Sorry we ran out of time there, but like I said, a lot to unpack and talk about as far as this topic goes. And with that being said, we'll conclude things and really appreciate you tuning in for this topic. I know it's a very tough time as far as fertilizer prices and volatility. And of course, soil biology is one of those many tools out there that can provide insights on improving nutrient use efficiency, understanding maybe where you can make fertility cuts. Like I mentioned, related to nutrient consumption and immobilization, you can prioritize which fields maybe have the least risk. And then, of course, all the great solutions out there as far as biofertilizers, biostimulants, enhanced efficiency fertilizers, a lot of great insights you can gather on targeting and placing those solutions as we just went over. So thank you all. Really appreciate your time and have a great rest of your day. Thank you.